When we talk about water and agriculture, definitely we also consider what we call it virtual water. When we import, we trade on grains, we take water from other side when we import and we bring it to us. Israel has a huge component of bringing, importing wheat, and the relationship, I don't know if you ever know that for each ton of wheat you import 1,000 tons of water. So also is an element to be considered when we talk about water and agriculture. Our next speaker is Assaf Distelfeld. Assaf is a staff member of the Department of Molecular Biology and Ecology of Plants of Tel Aviv University. His laboratory is in the Institute of Cereal Crops Improvement, and he has been focusing on trying to improve yield and quality of crops, especially on wheat. And we're looking forward for the last talk before lunch. So I will bring you some water.
Yeah, so I'd like to thank the organizer for inviting me, but complain about the time slot, which is a bit problematic, but at least we know the problem now. And the problem, of course, is food, or that we have a system biology. Yeah, our system biology is speaking right now, and the solution, of course, is it's also simple. And the solution is to have more food. But how we can achieve that? So, of course, I am talking from the breeding point of view that increasing the quality and yield of wheat can be achieved by the introducing and combining of beneficial genes or allele. And wheat produces 20% of the world calories, and this is why it's important to invest in wheat. And so where can we find those beneficial alleles? Part of the answer can be found in the evolution and domestication of wheat, which I will briefly... Oh, we have a water supply. So it can be found in the wheat evolution, and I will briefly go into that. It's 500,000 years ago, there was a hybridization of two diploid wheat-related species that gave rise to wild emmer wheat. This is wild emmer wheat that was domesticated 10,000 years ago and gave some tetraploid wheat. Now it's a tetraploid because it's a combination of two diploid species. And this is durum wheat that is we are producing pasta out of it. And it was later in cultivation, it was hybridized with another diploid species and gave hexaploid wheat, which is used to prepare bread. And this is 95% of the wheat that is grown in the world. So if we think about from a genetic point of view, we have genetic diversity in all those levels. And the genetic diversity in the diploid species and also in wild emmer wheat is twice as much as we have in cultivated wheat. And what it means by genetic diversity is that we have a lot more combinations that we can, more genes, more alleles that we can find there, and combine them together to have a better yield and quality in wheat. And this idea is not mine. It's originates from the discoverer of wild emmer wheat, Aaron Aronson, that he has a vision that it will revolutionize the wheat production. I don't know if it will create a revolution, probably not, but I think it can improve the yield and quality of wheat, and this is a strategy that I'm working on in my lab to study that. And I will first talk about the quality of wheat, which is mainly its protein content. And previous studies that also were developed in the Weizmann Institute by Moshe Feldman and Avi Levy found that wild emmer wheat, which is this beautiful spike here, has at least twice the protein content compared to cultivated wheat. And the question as a geneticist is, what are the genes that control this high level of grain protein content, which we call GPC for grain protein content. And the way to start to answer that genetically is by doing crosses between, like we see here, for example, between cultivated wheat that we have to emasculate to take out the anthers and wild emmer wheat. And then we take the anthers from wild emmer wheat, put them on the female flower of cultivated wheat, and if all goes well, we have hybrid seeds, and we can later produce genetic populations that are segregating for the traits that we want to study. In this case, we want to study the grain protein content. So such population was from a dicocoides, from a wild emmer wheat that was collected in Israel by Moshe Feldman. He sent this wheat to a researcher, Joppa, in North Dakota, and he developed a mapping population, found out that QTL is a chromosomal region that is affecting a trait, a quantitative trait like protein content. He found out that the QTL exists on chromosome 6b, a pretty major QTL. And when he retired, he transferred this material to George Dubkovsky from UC Davis and Sion Fahima from the Haifa University, and they got a BARD grant to clone this gene, to find out what gene is responsible for this QTL. And then I joined into this project as part of my PhD thesis, and what we found out that it's not only protein content, but also zinc and iron. And we heard in the previous conversations that micronutrients are very important for human nutrition. I also want to mention that in addition to importance for human nutrition, the protein content is also important for industrial uses, what you can do out of wheat. And in UC Davis, Cristobal Wawi, that was the parallel PhD student that worked on that project, found out that this QTL has an effect on senescence. You can see here, senescence is when the plant is getting old. Like you can see here that when we have this allele from the wild, the plants senesce faster compared to cultivated wheat. So what could be the mechanism that connects all those findings? What we know that until flowering time, wheat absorbed most of the minerals, most of the nitrogen from the soil. And then the amount of minerals that we are getting in the grains is a game of how much the plant can remobilize from the senescing leaves to the grain. And it seems that this gene has to do something with this process of remobilization, of how efficient the transport of minerals would be from the leaves to the grains. And then the task, the objective was to clone this gene, and I'm not going into details about that, but just to say that the technology since 2004 or 2002 that we started this project developed a lot. So today we are probably would have been able to clone the gene even faster than we did. But also back then we used a principle of collinearity to get closer to the gene. So before we started to use this system, there were only 2 molecular markers in the region. And molecular markers are helping us to get closer to, they are improving our resolution and helping us to get closer to a gene that we are looking, that is responsible for a desired phenotype. And we used the rice sequence genome to find out the genes that are in the region, and then developed markers to get closer to the critical chromosomal region, which is responsible for the high grain protein content. And today there are wheat genetic maps that have thousands of markers, and this is a technology that already exists. And after we saturated the region with the markers based on the collinearity, we did the next steps of building the physical map of the region, which is actually the DNA sequences that are responsible, that are in the critical region, and found out that the gene that was responsible for the QTL we found out that there was one gene that was responsible for the phenotype, and then we were able to answer the basic question what are the differences between the wild and cultivated wheat in relation to this gene? I mean, what are the different alleles? So this is how genetic diversity looks like. When you sequence the gene, you can see that in the cultivated wheat, we have an insertion of additional nucleotide, additional letter in the DNA code, and this insertion disrupts the protein, and this is why we are not getting a functional protein and less efficient process of remobilization of minerals from the leaves to the grain. And this gene is a NAC transcription factor that is specific to plants. You cannot find it in humans. And what we did next is wanted to see its expression pattern, or where this gene is active, because we know that not all the genes are active all the time. And then we saw that the gene starts to express, to be active around flowering time, and goes up all the way through the grain filling period, through the senescence. And this correlates well with the phenotype that we saw that has to do with the remobilization of minerals from the leaves to the grains. And you can see here 3 lines, and not only one, because wheat is a polyploid species like I showed before. So the 2 other genes are the homologous genes, or similar genes that are found in the different genomes of wheat, and they seems to act the same, or at least to be expressed at the same pattern. And this is an image that I found today actually in the web. This allele was introduced into cultivated wheat and in a breeding program of George Dubkovsky in UC Davis, and those seeds are commercially available. Seeds with high protein content that is caused, elevation of 10% in the protein content that is caused by this allele from wild Emmer wheat that's originated from Ramot Naftali in Israel. And just to conclude, this is concluding nicely this story of the quality of wheat. And then I want to talk now about the work that I did here. We did here in my lab. And like I told you before, there are 3 homologous genes, and we wanted to ask what is each gene is doing. Maybe if we can modify also the other genes, we can get better quality or other phenotype. And to do that, we used a technique called EMS, stealing mutation. You can treat a plant or seeds with a chemical that does changes in the DNA, creates mutations, and this is artificial allelic diversity. And then, because we know the sequence of the gene, we can isolate the mutants for specific genes. And like we did here, we isolated mutants for every copy of this gene. And then we asked, what is the phenotype? And we saw some phenotype in pod experiments, but of course when you grow wheat, you want to see the phenotype in the field, because this is the real test to see a real phenotypic effect. And we planted the mutants in the botanical garden. We have a small field plot over there. And the plants grew, and you can see here that there are no differences between the mutants. And this is expected because, as I showed you before, the expression pattern of the gene, the gene only starts to express after flowering. And here, this is flowering time. The plants are uniform, and there are no differences. as expected. But later on, we were able to see that the mutants, they show delayed senescence compared to the sister lines that have the functional genes. And when we ask the question, and you can take a closer look at that, it's a very clear phenotype. And we ask, we measure these differences in senescence by measuring the chlorophyll with a simple equipment, and we were able to see that there are significant differences between the double mutant that had the most severe delayed senescence compared to the wild type, and here are the single mutants of the gene, one in the A gene and one in the D gene of wheat. And this was also mirrored in the protein content that was much lower in the mutant, and here the double mutant has 15% less protein content than the wild type. So the idea wasn't to make wheat with a lesser quality. The idea was to ask what is the plant is doing in the meantime. When it's green and nice in the field, is he still doing photosynthesis? Because if the plants are doing photosynthesis for a longer time, we're thinking that maybe we'll get more yield out of the field. And we measured the photosynthesis, and we did see that those plants are doing photosynthesis, and there is a chance for a higher yield. But then we checked it's the spike dry weight throughout the senescence, and we saw actually no differences in the spike dry weight, and also we measured the grain weight, and also no differences. So we asked the question, what happened here? And maybe a possible explanation is we can learn from overlaying the two graphs of one of senescence, okay. And you see here the senescence, those are the wild type, and this is the double mutant that has delayed senescence. And this is the gain of weight in the spikes. And you can see that the two processes of senescence and grain filling periods are very correlated. They are ending actually at the same time. So what we did here is actually changing one element out of two. We changed the element of the source. We produced a source, we produced leaves that can produce carbohydrates for longer time, but we don't have the sink capacity to use those carbohydrates. This is our hypothesis, is that we also have to change the sink capacity. Which leads me to the next topic that I have 3 minutes, I guess, to talk about is wild emmer wheat can help us to increase the yields, the sink capacity of cultivated wheat. And this is not trivial, because the grains of wild wheat are much lighter than cultivated wheat. Okay, sometimes cultivated wheat can be 4 times heavier than wild wheat. It's like if we compare to humans, it's like marrying a gorilla and hope that your sons will be Einsteins. But good. Okay, she's expensive, but good. Okay. But fortunately, in plants we have a lot of offsprings, right? So we can do this experiment a lot of times. And this is what we did here. You see the field in Emek Israel that Debbie mentioned yesterday. And we have 500, a progeny of 500 offsprings that are actually backcross plants. You know, in plants you can make a hybrid and then backcross back to the cultivated, in this case to the cultivated parent, so to get only a small piece from wild wheat. And then you can ask, is this small piece is not dragging the unwanted traits, but only beneficial traits? And this is what we ask here. And we were able to see, to identify 15 lines out of 500 that showed a significant grain weight increase. And now we wanted to study it more carefully, and we aligned here. This is wild emmer wheat, and this is the cultivated wheat, the two parents of the population. And you can see the color differences are because of the protein content, but this is from the previous topic. And you can see here how domestication worked. The domestication worked on increasing the grain width of wheat. And what happened to the grain length, it actually decreased. And this is something that we see not only in that population, in those two specific accessions, but also in other reports in the literature shows that. Okay. And so let's see what happened in our lines that show the increased grain weight. We can see that, yeah, we have some increase. I'm not betting on it that it's a significant difference. But the real story is here, that we were able to get lines out of this progeny that has restored the length of the wild emmer wheat, and as a result were heavier than the original cultivated wheat. So this is an increase of around 7%. This is again like in relation to the previous talk, it's not an exponential growth, it's more of a linear growth, but this is what we can do, I think, in plant breeding. There aren't any silver bullets, but there are beneficial alleles that we can identify also in wild emmer wheat. So we map this genetically to a chromosome using a genetic map with a lot of markers, and this is a collaboration with Jan Dvořák from UC Davis. And now we even have a better platform for doing this genotyping, and we hope to use it soon and repeat this experiment for we are repeating it for this year. This is from again from the VAR in Vulcani Institute in the north in Emek, Israel. And of course, we have to repeat it at least one more time to see that the results are reliable. And if they are, we are going to target this QTL and try to identify this gene. And I would like to acknowledge my department, the ICCI, and my lab members for supporting me. And I'm the new guy here, and I feel like home, so thanks to them. And for the funding agencies, which are the BARD and the Marie Curie reintegration grants. So, thank you.